The more we know about how RNA functions, the better we can develop RNA-based therapeutics and vaccines. But just how do we analyze RNA? Enhanced cross-linking immunoprecipitation, or eCLIP for short, is a state-of-the-art method to map RNA binding sites across the transcriptome. Developed less than a decade ago, eCLIP has provided insights into RNA biology to hundreds of researchers and drug developers. In this inaugural episode of RNA Insights, we talk with eCLIP co-developer and Eclipsebio co-founder Dr. Jean Yo about his career combining computational research and biology with the advancement of RNA medicines through improved RNA analysis. Welcome to RNA Insights. Thanks for meeting with me today. Just to get started a little bit more about you and your career, how did you get into molecular biology and RNA biology initially?
Yeah, that's a good question. So I had almost zero molecular biology as an undergrad. I was studying in the areas of engineering, economics, much more focus on thinking about technology. When I decided to go to graduate school, I also didn't go to grad school because of molecular biology. I went to grad school in the neuroscience, the brain and cognitive sciences department at MIT, and it was to really think about and learn neuroscience. I realized that systems and computation and neuroscience was very exciting, but for me, it was very difficult to go from prediction to validation very quickly in terms of the models for neurodevelopment at that time. And so I then started gravitating towards an also emerging field then, which is genomics. The human genomes were just recently published, and the draft human genome, and then the mouse genome. And so I was excited about that potential because the machine learning or the computational technology methods that I had learned could be applied to this, vast amounts of sequence data. And so I joined the lab. I was co-mentored by Chris Burge, who was a fellow with Phil Sharp before.
Dr. Phil Sharp discovered RNA splicing and won a Nobel Prize for his discovery in 1993. RNA splicing is the process of removing non-coding regions of RNA, called introns, from precursor mRNA. This leaves behind only the coding regions, called exons, that form a protein. Another form of splicing is alternative splicing, which is when RNA is spliced in different combinations of exons to create related but different proteins from RNA. These proteins are called isoforms.
And so we together started thinking about how do you understand RNA regulatory elements and how they control alternative splicing. And at this point, express sequence tags were used to identify different alternative isoforms. And so I ended up being forced to really understand molecular biology from the conceptual side, but also on the experimental side. The molecular biology perspective was a somewhat random walk into that space there.
It seems like a lot of scientists have a similar idea of, oh, I went in with one thing and came out totally different. Did you always want to be in the lab, even if you didn't know necessarily that field? Or did you know what you wanted to do when you initially started your grad program?
I had no idea, right? And so I was just interested in learning more about life. I wanted to come with the perspective that an engineer and more quantitative person had towards biology. And so I sort of meandered again to neuroscience because there was a fair bit of computational analysis and it's sort of a blend between machine learning and then thinking about data. I would say if you ask me, I don't know, many eons ago, whether I knew I would be an RNA computational biologist or experimental biologist or working in neuroscience or neurodegeneration, I would have said I would have no inkling that that would have been my eventual career. I'm not sure if it's lucky or not, but it's just ended up that way.
Once you did end up going that path and getting really into the biology and wanting to go that route, you said you were talking with your advisor and starting to have these questions. What were some of your first steps to pursue answering those questions?
As a graduate student with Chris, many of the projects that I
Mm,
got really interested in was around the central idea that there were hidden signals in the RNA or the transcriptome that would control RNA processing. And at that time, this is prior to Celexa or Illumina sequencing. We're limited by the availability of cDNA or express sequence tags that were publicly available that we could download and then that we could then align to the genome and identify different isoforms. And then that formed the initial basis for, oh, look, there are these alternative splice isoforms, what controls them. And there was a flurry in the field, and I was involved in this to try to find elements that could control, enhance splicing, suppress splicing, modulate the utilization of different splice sites. And so I did a fair bit of splice site modeling. That was my first paper in Chris's group to understand what makes a splice site stronger or weaker. At the end of it, I was excited that some of these rules, the computationally defined sequence features could actually reasonably predict conserve alternative splicing, things to conserve between human and mouse. So that was my final publication at MIT.
I've linked to these papers called Maximum Entropy Modeling of Short Sequence Motifs with Applications to RNA Splicing Signals, as well as Identification and Analysis of Alternative Splicing Events Conserved in Human and Mouse in the show notes. You can also access these and Yo's other papers from his lab's website, which I've also linked in the show notes.
Then after that, I realized that it would be interesting to see these variations in RNA processing, right, splicing included, in different cell types. And one way to go would have been to get a lot of human tissues and then sequence ESTs and all of that. But again, this was slightly before these commercial instruments, sequencing instruments became mainstream. And so at that point, people leveraged splicing sensitive microarrays. And I started developing ways to analyze these arrays. The previous methods were not really ideally suited for splicing analyses. But then we would apply RNA from cells, and I wanted a system where I could study many different cell types with the same genetic background. And I gravitated towards maybe embryonic stem cells could be a good model because you can take an embryonic stem cell, indefinitely proliferate them. At that point in time, it was difficult to work on embryonic stem cells in many parts of the country, at least to get federal funding for it. And so California had this initiative to fund embryonic stem cell work through California Institute of Regenerative Medicine. And so I ended up deciding to move to California. I worked with Rusty Gage because Rusty was one of the eminent scientists that studied and discovered neural stem cells. And so trying to take embryonic stem cells and differentiate these into neuronal stem cells and then into neurons and so on was a major bottleneck at the time. So I basically did that move to have a system to then study RNA biology. And I did. I applied RNA from embryonic stem cells and neuroprogenitors made from these stem cells. At that point, it was still very difficult to do these experiments, put them on these splicing microarrays, and then identify alternative isoforms that were controlled by regulatory elements that resembled sort of the elements that were bound by particular RNA binding proteins, like the RNA binding protein, sort of the RB-FOX2 family. That led to our continued interest in now studying the trans factors or the trans regulatory factors that control RNA splicing and then eventually other aspects of RNA biology. Around that same time, the Yamanaka factors, right? So Yamanaka discovered or invented the process of reprogramming. And most of us gravitated then to leveraging this process to generate induced pluripotent stem cells or iPS cells from fibroblasts or from more mature cell types as a way to generate an embryonic stem cell system.
In a bit more detail, Yamanaka factors are four transcription factors that activate a gene in adult cells to reprogram them to act as induced pluripotent stem cells. Once reprogrammed as stem cells, these iPS cells can be used in the same way that embryonic stem cells would be used for researching many cell types.
And so we were now able to apply for NIH funding to work on iPS cells, and because they're no longer derived from the embryo. And so now the iPS field became a thing. And so my lab, when we started at UCSD, started working a lot on iPS models for neurobiology and neurodegeneration. But at the same time, I wanted to scale the process of studying RNA binding proteins, which are the trans factors that regulated splicing and other aspects of RNA processing. So we wrote this
Really?
very ambitious grant at the time to try to map all the RNA binding protein binding sites. And anyway, that sort of led to eventually technologies that we deployed within Eclipsebio.
I definitely want to break down some of these technologies, but right before we get there, that's a lot of challenging things happening at once, going from MIT and trying to find the right medium to do your research to moving to California. Did you experience a lot of challenges with that or even pushback within the field of trying to use embryonic stem cells for your research, especially if some states I know at the time were iffy on it, or was that a pretty smooth process?
The move to California was for me the right move because when I got here, I think many of the scientists viewed stem cell research very positively because it was a way to understand human diseases using human cell types that you can derive from sort of a clean slate from the epigenetic perspective. Personally, I was very happy and excited when the IPS field came around because then that expanded the ability to understand the impact of genetic variation to say neurodevelopment, neurodegeneration, and or whatever cell type you can imagine, because now you can get patient specific fibroblasts or blood cells and then reprogram them back into an IPS stage and then generate neurons and other cell types. But now the cells harbor the variant or the mutation that was present in the patient. And this technology Yamanaka had invented really opened the field for development and for disease specific research. I was much more excited about all these opportunities than I was in paying attention to the challenges there. There's just a lot of things that now scientists could do. I felt very lucky because I started my lab in the confluence of at that point next gen sequencing, right? So Illumina, Solexa, we had access to all these next generation sequencing technologies that became a sort of sequence-based digital microscope into studying genome variation, RNA variation. And then at the same time, there were these big efforts in trying to understand how you make different cell types from iPS cells, more and more sophisticated ones. And now many groups, not ours, have also invented other more sophisticated models for human development. And then at the same time, there was a booming interest in computational methods applied to biology. So there were more and more folks that were physics trained or computer science trained, and they were entering biology. And so there was this common excitement about can we apply these methods that was traditionally being used in the image analysis of vision and speech recognition, and now applying that to biology. And I would argue many of these advances that started, I think, early 2000-ish have progressed to now. I think there's still a lot of excitement in, now you have AI, right? I mean, I would argue machine learning and AI are synonymous, but okay, fine. There's like large language models that are popular now. And so that still represent the field of machine learning and computer science. And then there is still a lot of work being done, again, by many other leading labs in developing organoid models for brain development and other more sophisticated models for cancer using patient material that you have now generated into a self-renewing population. And then, of course, the RNA world has just kept expanding. It's been great to sort of see this evolution across the last 20 years.
For sure. RNA obviously has boomed in the past couple of decades. But a lot of that also, I feel, had to do with what you were doing at your lab and some of these technologies that you were referencing. Obviously, one of the main ones being enhanced cross-linking immunoprecipitation. Do you want to talk a little bit about developing eCLIP and developing that technology?
Right. I think the impetus for this was the idea that we wanted to map the regulatory elements bound by many RNA binding proteins in human cells. I think the grants that we put in may have been a bit more ambitious than we had thought. So I had at that point applied Eclipsebio, right, or HitsClip, which was originally developed by Bob Darnell's lab at the Rockefeller Institute. And we had applied it in 2008 or 9, and then we started applying this to a family of RNA binding proteins known as the HRMP family of proteins. And I think we got overconfident. We're like, oh yeah, we are getting good at this technology. Let's apply these to hundreds of proteins. And then we face many challenges. We realized, okay, most of the commercially available antibodies for RNA binding proteins were not great. And so then we had to launch an effort to identify and characterize in a systematic way using pretty rigorous methods hundreds of commercially available antibodies and some not even commercially available ones. But we had sort of developed partnerships with many biotech companies early on to absorb and evaluate all these antibodies. So we spent about 2 to 3 years basically just doing characterizations of our
Mm.
antibodies for the ability to IP or immunoprecipitate an RNA binding protein RNA complex. And then we realized even with these antibodies, because they vary a lot in quality, the traditional Eclipseq or crosslinking IP sequencing method was not as easy to deploy or robust for us. This particular method required UV crosslinking, you cement the protein RNA interactions, you digest away unprotected RNA, and there's a series of molecular steps that then eventually leads to a DNA library that you can plop onto the sequencing instruments.
So, as Yo said, traditional CLIP involves two main steps. In the first step, cross-linking, researchers expose RNA to ultraviolet light, which pauses all protein interactions on the RNA and leaves the RNA as a snapshot of the interactions that were occurring at the moment the RNA was exposed. Usually, researchers then fragment the cross-linked RNA to perform the second main step, immunoprecipitation. In this step, researchers isolate the targeted proteins in the RNA by using a selective antibody that binds to that desired region. Once bound by the antibody, researchers wash away the rest of the RNA, leaving only the targeted proteins and RNA behind. Having the correct antibody for this process is critical.
At that point, it was early days as well, so the reads were shorter, they weren't as abundant, and so being careful about how you generate the material so that you have enough material to sequence and that the diversity of the molecules were large enough that you can derive statistics from the data. That ended up being a more difficult thing to do than we had imagined. I had thought this is my lab being young in this area, so we just maybe didn't know how to do this correctly. But then when we asked around, this was a major problem. And so the original developers of the technology for the CLIP technology at Rockefeller also had made other advances to improve the efficiency of technology. And we spent a year running hundreds of experiments with these updated methods. But again, they were very sensitive to the quality of the antibodies, the choice of RBPs. And so then we got worried that we will not be able to complete our mission in the grant. At that point in time, I think Eric Van Nostrand, whom I've known since he was an undergrad, was a postdoc in my group. And then we had folks like Alex Trishkin, who had then spent some time in my group. And they, together with a team of technicians, sort of spend a fair bit of time trying to understand all the different pieces and steps in these traditional protocols and just trying to enhance every little bit of it. So I don't think I can point to like a single place where suddenly you fix this, the protocol is better. But I think there was a fair bit of optimization. And so the enhanced CLIP protocol as an optimized version of what we had to do for our ENCODE project, which is encyclopedia of DNA, elements, including RNA elements.
The ENCODE project was established by the National Human Genome Research Institute in 2003 with the aim to help map all functional elements in the human genome sequence. Yo helped map numerous RNA binding proteins for ENCODE, and eCLIP helped him and others characterize RNA to locate RNA binding proteins efficiently and at a much larger scale. More information about the ENCODE project is available in the show notes. In addition, you can find Yo and Nostrand's original paper on eCLIP in the show notes.
So I looked at it as we needed to do this for this scale of project. We couldn't spend years characterizing just one antibody and one RNA binding protein. We had to, spend a short amount of time doing hundreds of antibodies and hundreds of CLIP experiments. And as a caveat, there were always some loss. We couldn't do as many replicates as we wanted from the sequencing costs that we had for early instrumentation. We could have definitely optimized some of these digestions for individual RBPs. But overall, I'm pretty happy now with the initial draft of these data sets. They've all been leveraged by hundreds of labs and many companies, including biotech companies as well. We view the project as a great way to lift the field of RNA binding protein RNA biology, because now folks that were not familiar with this area, especially from the computational side, could dig in the data. And folks that were thinking about this problem from the genetics or cell biology side could now understand what examples of this data could look like and how that could help them understand their specific biology.
Correct me if I'm wrong, but it seems this need and interest from so many different people is what spurred the opening of Eclipsebio as a mechanism to help share
Yep,
that data.
that's right. So even prior to the eventual publication of Eric's paper in Nature Methods, demonstrating that the eCLIP method could really scale. It had high diversity. It had much lower failure rates. Even before the paper was published, we had sent the protocols out to anybody who needed it or wanted it. And when the paper was out, we had probably 1 or 2 emails every week. Well, I got emails every week about some lab that wanted to leverage the protocol, and I try, as you know, I'm very collaborative, so I try as much as possible to help as many friends, and colleagues as I could. But at some point, it was just not scalable for me because I had my own NIH money, I had my own CERN money, I had my own discretionary funds, but we were just trying to help others solve their problems, and we had other own problems to like look at and be interested in. So at that point, I think Peter Chu had approached me because he had talked to the licensing office at UCSD and was interested in technologies that the lab was leveraging and applying to solve problems in biology. And I said, I just told him like this is a major bottleneck for me because I would love to help other people, but I'm limited, right? I'm running my lab and I'm solving other problems and writing other grants. And so together with Eric Van Nostrand and Alex Shishkin and Peter and myself, we said, well, maybe we should just have a small company that could make this protocol even better in terms of industry sort of optimization. So it's more robust, it's a better protocol, and then have it then be applied as a service to whoever needs it. So I was initially thinking that this would be a good way to enable the academic community. And I think we did that for a fair number of years before we realized that the academic community, although vast, individually did not want to spend, did not appreciate the amount of effort that this protocol would take. And so they also didn't want to spend the resources. And so the academic market was great, but then Eclipse had to pivot to many other assays to build an entire ecosystem for technologies that probe RNA. And then with the growing excitement in the siRNA space and the ASO space and the mRNA medicine space, we had to deploy our tools at Eclipse also differently to different problems. Where I think Eclipse started was as an effort to enable the academic community. Where it's at now it's now a company that catalyzes RNA-based therapeutic research and the discovery of new drugs by pharma, biotech, and also the academic community. That journey was a long one. I don't take much credit. I think I helped, but it's been a team of people, and especially with Peter and others full time at Eclipse that has gotten us where we are.
Talking about this growth of technologies and expanding, I know throughout this interview, you've alluded to how sequencing becoming more and more mainstream has impacted RNA so much. Is that process still happening, would you say today? Especially, like, I feel like in Eclipse Bio, you can see that, or in other RNA therapeutic companies and research.
Yeah, I mean, sequencing has become a low cost microscope, right? For
Mm.
everything in molecular biology and cell biology. I mean, even in directly to drug discovery, it has gotten us to develop as a field to develop new ways to leverage sequencing to understand how to perturb biology as well. So, for example, yes, you can sequence RNAs, you can sequence protein, RNA interactions, you can sequence small RNAs, and all these, features. So Brian Pagel from UC Irvine came yesterday to speak, and he spoke about his efforts in using sequencing to do small molecule drug screen, right? Because you can conjugate or associate a small molecule that you can make as a library to a DNA sequence attached to the bead or to the molecule, and then you can do screens that way. So sequencing also enables us to identify small molecule interactions. Others have done use sequencing for antibody drug discovery. It's been an amazing technology set, and there are many companies in the space. There are also long read companies like PacBio's efforts and Nanopor's efforts to, at least for me, help us identify isoform specificity and RNA modifications. And I think there's just going to be more and more of these technologies that will enable molecular and cell biology.
Goes well into my next question is gonna be, what do you think will be the next, like, big thing in RNA therapeutics? I know you mentioned AI earlier as well, and as we've been talking about, especially since the pandemic, just RNA therapeutics in general and vaccines especially have just blown up in the past 5 or 6 years. As that research continues, what do you think will be, like, the next big thing?
I think RNA as a substrate and a target for reagents, molecules like sRNAs, ASOs, even small molecules, RNA targeting proteins and so on. That's very well established now that that works. And so there's more and more modalities that will appear to now leverage the RNA and in the cell and be able to have disease modifying effects. And on the other hand, mRNA has or RNA has a drug itself. So the mRNA protein replacements, cancer vaccines, so on and so all these all have the concept that you provide exogenous mRNA and then they get translated or processed and then enable the cells to do something different. I think that's it's gonna keep going, right? I think folks are gonna leverage these technologies to be able to reprogram cells in the person, not just to make in vivo CAR T, but also maybe rejuvenate old neurons into younger neurons or old cardiomyocytes into younger cardiomyocytes, be able to provide a replacement. And now I would argue all genome editing technologies leverage mRNA medicines, mRNA technology. But I think there's a lot more to be done. I think the place where we are particularly interested in is can you do more with RNA? Can you basically develop other tools with RNA that is not just producing a protein to have some sort of effect on the cell? Can you keep leveraging sequencing to understand other aspects of RNA biology that has traditionally been ignored because of the biochemistry limits or that was just something missing that will enable higher resolution. And so we've been particularly interested in that area in ability to get single cell or spatially resolve measurements of mRNA, not necessarily translation, but sort of the combination of translation and half-life measurements. So things like protein output, but from an RNA-centric perspective, I think there's a lot still to be done. We're interested in also trying to understand there are all these RNA modifications. Do they all matter? How do they influence neural development? How do they influence neurodegeneration? How do they define genetic variation between individuals across large populations and rare diseases? So there's all the problems that were started to be interesting, I think 20, 30 years ago are still very interesting and not all of them are solved.
For sure. It's a great space to be in right now to keep going. What would you say in your work of RNA biology research, what do you think is the most challenging part of your career?
I think the most challenging thing these days are to align opportunity with the right level of motivation and resources. And I mean, that's not a new thing. It's always been a challenge. You want to chase a problem down, but you normally have to have the right person in the right environment with the right resources to do that. I think Eric and Alex and the team of technicians were really special and fundamental to the development and improvement of these technologies for scale. And I'm not sure if a different team that would have happened, at least in my group, maybe other groups. And so a continued challenge for me is to identify people, the right people that can solve a specific set of problems and just give them the right resources. So it's not a new problem or one unique to me. I think it's a one that we are continuing to face, particularly in light of more difficult federal funding, in the light of a decrease in international interest from fellows to come to the U.S. And just more skepticism and science from the public. I think all that adds some headwinds to really cutting edge science and innovation. That's become a challenge in recent years.
It is such an issue, and we're hoping that things like this podcast and ways to communicate science are gonna help show science is cool, science is so important. We need this for people's health. On the flip side, though, ending on a more positive note, what is your favorite part of your career?
I think working with the students and the postdocs and the technicians in the lab have been the most fun part because I am often very pleasantly pleased and surprised by the level of ingenuity and dedication that the trainees have and display in their work. Whenever I start a project, I don't always know where it's going to end up.
Mm.
And for many of the projects, not all, I continue to play a role in which I try to direct the outcome of the project in the sense that I am helping the student navigate through his or her challenges from the project. I'm like, well, maybe you can try this experiment. Maybe you can use this new technology. Maybe you can collaborate with this other group. So I spend a bit of effort doing that. But every so often, a person comes to me and has found a problem, solved the problem in ways which I would have never imagined. That is, for me, very exciting. I think as a scientist, I'm always intrigued by and curious by not just the outcome, but also the process in some sense of how someone achieved that outcome. And so the trainees have been, I think, amazing for the most part. And just watching where they go. We have folks that have now gone to be CEOs and CSOs of their own companies or other public companies and private companies. And so that's amazing to watch. And you can see in their career, many of them still have an RNA basis, a disease focus. And a lot of that, it probably came from their interactions with people in the lab, their experiences in the lab. Of course, they have all gained many more experiences in the real world in some sense, but you can still see like what they're doing. They're still, you can pull a thread to their journey, and some of it still has an origin to what they have learned in the group. So that's very cool to see. And of course, some of our alumni have also gone into academic positions, and you can sort of see them flourish, take what they have accomplished here in the group and have expanded or change directions, but you can still see this RNA focus and interest in diseases, technology development that is still inherent in them. Many folks have gone off to become group leaders in nonprofit or for-profit companies now developing next generation RNA therapeutics. All students have gone to amazing jobs in biotech and pharma, and all are leveraging what they have learned, but all obviously learning more. So the lab feels to me like a family, and when people leave the lab, they don't necessarily leave the family. They just go off and do the next thing. For many of us, I still keep in touch with folks. We remember their birthdays. We try to stay apprised of what they are doing. Of course, a lot of folks are very busy. They keep moving forward, but I try to get the folks that have gone away and gone on to try to help advise the junior people in the lab, and so it's self-sustaining mode of experience sharing in some sense. And that's just great for me to watch, to see someone that has gone from a trainee to a mentor. And I think that journey is a fun one, right? So that's the fun part of being an academic person. And also I see a lot of my students have gone out to start companies with us or join companies that we have started, the Sanford Laboratories folks and the other companies we have or nonprofits that we have helped to originate are like providing a great playground for innovation. And so that's been fun to have. I feel like have done my role as a mentor. I think my academic career is fun, but my academic career plus these other commercial ventures basically expand again our family set that again keeps folks in close interaction. It's always nice to have this idea that when your kids grow up, they still come hang out. So now, like, in my mind, the Eclipse folks are also part of my broader family. I think looking at where we've been, it's been an amazing progress and evolution of the company. And in some ways, the company has changed a lot more than a normal academic space. Obviously, it's not an academic lab, but the progress and the level of professionalism, it's been amazing to watch.
Thanks again to Dr. Jean Yo for joining this episode of RNA Insights. Before we leave, here are a few pieces of RNA therapeutics industry news. Circio initiated a technology evaluation agreement with Acutus Therapeutics for in vivo CAR T cell therapy. The agreement aims to see if Acutus LNP delivery that targets T cells can improve the stability and efficacy in vivo of Circio's circular RNA therapeutic called CircVec. Afrigen Biologics announced a collaboration with ResIP, an affiliate of Forge Bio, to develop an affordable vaccine for RSV. The partnership combines Forge Bio's antigens with Afrigen's end-to-end mRNA platform. Imorna announced clinical data for JCXH213, an investigational in vivo CAR T therapy for cancer and autoimmune diseases. The data reported that the therapy depleted B cells and blood and lymph nodes in the first patient with systemic sclerosis to be dosed with the therapeutic. Thanks again for listening to RNA Insights. We'll see you next month when we talk to another RNA expert.